Hello, this is the next in the series of videos for biochemistry. With that, so for this one, we're going to skip ahead to the intro to metabolism. With that, so this is going to get us started on to the rest of the course, which will end up being things like glycolysis and gluconeogenesis and citric acid cycle and how we make molecules and make energy. But so most of the things a cell does is it falls into two categories as far as metabolic pathways go, and it's either to create energy by using large molecules to grade down to small molecules and energy, or it's to build new molecules that the cell needs. So having these steps linked, these chemical steps either breaking down or building up, that's what's called the metabolic pathway. The energy currency for most of these pathways is actually going to be adenosine triphosphate, or ATP. So there's not really, as far as I know, any reason why it was ATP and not GTP or TTP or whatever. But ATP ended up being the one. And most of things funnel through ATP. Just because it can, it only has to, the cell mostly just needs to focus on building one major component. If you had separate ones for CTP and GTP and TTP and UTP, right, you'd have 5 different metabolic pathways you'd have to work with. And so what the cell does is says, okay, we're going to focus on ATP and use that for going to be able to build as our currency and create the other ones as needed, stuff like that. And so most of the power for the formation of these fuels is actually going to make ATP is actually through oxidation, and we'll talk about what that means in a minute. And there's really only there are a lot of different pathways, but a lot of the things, they're interconnected to give the cell flexibility, but also so that it doesn't have to make a new metabolic pathway from scratch. So it provides flexibility. So if it needs to shunt molecules from one pathway to the other to deal with different circumstances, it can, but also so that it doesn't have to build new pathways from scratch. But the other thing is these things are highly regulated, as far as because the cell doesn't want to use any more energy than it has to. And so if it has too much, it's going to have things in place to be able to stop it. If it senses that it's low on some component or it's low on energy, it'll be able to ramp up the production degradation as needed, stuff like that. And so these things are tightly controlled. Okay, so metabolism is just simply the overall process, and what's going to happen here is you're going to have several chemical steps linked together in a regular fashion to be able to go from overall starting materials to the final product. And if you have catabolism, so what you're going to do is you're going to start off with a large molecule and break it down into smaller ones and then into energy, stuff like that. So and then anabolism, that is where you take small molecules plus energy to make large molecules, and so it's just the opposite of catabolism, overall in concept, stuff like that. So if you're taking the lumber, the individual pieces of lumber, to plus energy, stuff like that, you can build a house with that. So that's all anabolism is. Catabolism would be to take the house and hit it with a sledgehammer. Not only do you go back to the smaller pieces, but also you release energy when you do that. So metabolism, right, it's highly interconnected, stuff like that. So you can sort of see the different metabolic routes and how they sort of fit all together. So lipid metabolism will be connected through nucleotide metabolism or amino acid metabolism, or come down here for other things. Something like that. And so all of these are interconnected. And if you sort of zoom in, you can sort of see these different chemical transformations, making GTP, ATP, deoxyadenosine triphosphate, so to formate the DNA pieces. So this is nucleotide and nucleoside biosynthesis, with that. So, but even then, this is going to be shunting in from different parts. So, by that, so where does the energy come from? There's two main sources. If you have the phototropes, so this is going to be mostly plants and photosynthetic fungi, like that. So it's mostly coming from sunlight. So we'll talk about that a little bit later in the course. Chemotrophs, this is where it comes from chemicals, essentially food, with that. And so you'll be able to get to that. We're going to be getting into that more in a bit. Okay, so metabolism is all about dealing with Gibbs free energy. So in the thermodynamics portion of this course, we talked a little bit about delta G. So the pardon me, delta G, stuff like that. And so so one of the equations, so you had the other one was, if you remember back, was delta G is equal to delta H plus minus T delta S, right? So like that. So that's one manifestation of Gibbs free energy. The other one would be this. So Gibbs free energy of the reaction conditions right now is equal to the standard Gibbs free energy under standard Gibbs free energy under standard conditions plus R, which is a constant, T temperature, and then the natural log of the products, concentration of the individual products over the starting materials. And so what you can do is you can actually, the cell doesn't really have any control over delta G, the standard delta G, the R or the T, right? You can't really change its temperature, stuff like that, but it can change the concentrations, stuff like that. And so by changing this, you can change the value of this to compensate to make this negative. So that, and so there's some things that the cell can do in order to be able to drive reactions. Okay, so one of the things it can do is it can link the reactions. So in this case, so if you really need to make C, just splitting A into component B and C, the delta G may be bad, stuff like that. But if you take B and then do something else to it that releases more energy, you can compensate by tying these two things together. You can make C, but you can go from A to C, but you have to link. But in order to get the energetics to work, you gotta do a second reaction, and that's fine. So the other thing you can do is you could change these concentrations a little bit. So that can help quite a bit too. You can either make this larger, which makes this overall thing more negative, or you can decrease the concentration of the products. So so if you think about reactants going to products, right? So you can sort of think of this as Le Chatelier's principle, where if you add a bunch of these, right, the system's going to go make more product. So in order to balance out the reaction. So that's what's going on here. If you make this concentration higher, you'll be able to. That makes this whole thing more negative, right? Makes this smaller, which makes it more negative, which can compensate for this. The other thing you can do is you can siphon away product. So you can decrease this concentration. You can either increase this concentration or decrease this concentration. If you decrease this concentration, you're decreasing this. Again, you're making this part smaller. When you take the natural log of it, it makes this whole thing more negative and can help compensate for a bad delta G standard. Okay. So so one of the ways that we can deal with this is through ATP hydrolysis, because this is incredibly, I guess the technical term is exergonic. You can also think of it as exothermic, to help drive the reaction. So if you want to just lop off one of the phosphates to go to ADP, stuff like that, so you can get out negative 35 or negative 30 kilojoules per mole. If you want to lop off both of them, you actually get out negative 45 kilojoules per mole. So for that, and the reason for this is that, and so this is typically the what we use. So with that, we can use the other ones, but this is the main driver for most cellular processes. If we need to make the other ones, things like GTP, CTP, UTP, whatever, Typically, those are actually made from the monophosphates, and then you just burn ATP, so like that, in order to be able to create them. So in this case, ATP provides an additional phosphate. So in order to go from mononucleoside or nucleotide to a trinucleotide, you would actually burn 2 ATP molecules, each providing 1. So now this one here, so you can think of it also as energetics. Some of that is equilibrium. So if you just had A going to B, say this is plus 16. If you tie this in with ATP hydrolysis, right, now the energy becomes much better. It becomes negative. If you look at the equilibrium constants here, it actually goes up 1,000-fold. So you can get a couple of reactions. So you can get a much higher. So I think this N, if I remember right, this is for how many ATP hydrolysis you have, stuff like that. So in order to be able to push the equilibrium. So why is it so energetic to do this? Well, one, you're releasing some additional resonance structures that possible for the phosphate. So like that, after you release it, there's more resonance structures possible. Also, you have this repulsion that can happen. So with it, and so you have negative charge, negative charge, negative charge, right? You have all those negatives right next to each other, and so being able to split that allows the repulsion to go away. So that's a big one. You have entropy driving this is 2. You're going from 1 molecule to 2, and even then you can even further hydrolyze this inorganic phosphate so that into individual phosphate molecule. Oh, well, actually, I'm sorry, this is inorganic. If you can also lop this one off to make another phosphate, and so that you can get a third molecule out. Also, you can get there is some stabilization. So if you have your phosphate here with a negative charge, and you have water, right? Remember, the water is going to have right. It has a partial positive here and partial negative here. So putting this negative next to this partial positive is a very good interaction. And so this, as you hydrolyze these, more of these interactions can happen. So to help drive the enthalpy of this reaction. Okay, so one of the things that the cell does is it doesn't always use phosphate or ATP. So there's sometimes where it needs to keep the phosphate there for later use, and so you can put it onto other high energy molecules like 1,3-bis phosphoglycerate, phosphatidylpyruvate, creatine phosphate, things like that. You can put these onto other energetic species. So like that for use later, or you can put them on lower energy species like that. So so what you want to do next is take and so this allows you to sort of use different things at different speeds. So if you have ATP, the energy can be released really quickly. So if you're a sprinter, right, you're going to be using mostly ATP. And then also these other high energy species, so like creatine. If you're running a marathon, right, you're going to start, your energy level is going to be aerobic and anaerobic metabolism is going to be which we'll talk about later. So like that, those will start to ramp up as being the important ones. So aerobic, right, you're going to get lactic acid production, things like that. So, but it allows you to do use different reservoirs of phosphates other than ATP. Okay, so in general, oxidation is going to be increasing the number of CO bonds with that. So, and usually that comes at the expense of CH bonds. Reduction is just the opposite. We're going to be reducing the net overall amount of CO bonds within the molecule. So with that, and also catabolism, right? It usually makes ATP. So oxidation of fuels is usually what drives this. So the overall oxidation for this reaction would be going from the aldehyde here with 2 CO bonds to 3 to the carboxylic acid. And so you can sort of see this. We reduce the number of CH bonds here by adding a new CO bond and going from here to here, something like that, and picking up. So this hydrogen here ends up making it an ADH molecule, which we'll talk about in a second. So this phosphate gets popped on here, and then we can use this to drive. This is a higher energy species than ATP, so you go up in energy. So like that, so you can go, well, where is this? You go up in energy if you can make this, and then going down in energy to make ATP. So that's okay. So we can use this to drive ATP production. But you do this, it comes at the expense of the oxidation of the carbon, going from 2 carbon oxygen bonds to 3. So the other way that we can help drive this is through gradients. And again, we're going to talk about this in a bit in later chapters, stuff like that. So either from carbon fuels to CO2 in aerobic metabolism, as well as producing ATP. So with that, being able to do this, putting protons in, taking protons out, you're going to get a net change in the number of positive charges on the outside, negative charges on the inside, and that's what help drives it. Okay, so phosphate stability. So we have this really high energy molecule. Why doesn't it just fall apart in water? Well, it turns out that this is a very high thermodynamically unstable. Right? You're going to lose 30 kilojoules per mole when you go from ATP to ADP. Stuff like that, but there's a high activation energy to go between there, right? Those phosphate oxygen bonds are going to be quite strong. Also, the negative charge that's on that phosphate is going to repulse any of the phosphate. It's going to repulse the phosphate. So getting water in here to go and do the attack is going to be hard because remember that it's this oxygen here that's going to have to come in and push away this negative charge. So it's going to be difficult for it to approach, stuff like that. And so in this way, it's kinetically stable because it has such a high activation energy. But what this allows you to do is turn it on and off with enzymes. So if you have an enzyme that greatly lowers this, you can regulate ATP hydrolysis very tightly through the making or reducing of the amount of enzyme that's in the cell. Okay, so overall, sort of for energy from food, right? You're going to degrade the small molecules, whether it be lipids, polysaccharides, or proteins. They're going to have their own sort of processes, but they are going to funnel down into a common intermediate, and in this case, it's acetyl-CoA. This can then get funneled into the citric acid cycle, stuff like that, and then so with that, the electrons from that will get pumped into the oxidative phosphorylation process, where it converts oxygen into water and then ATP. So with that, and so even though you have the first part of this is the same, stuff like that. So this allows flexibility by funneling everything in here into the same process, so you don't have to build completely separate pathways for the proteins or for polysaccharides or lipids. You only have to deal with this first stage, getting this stuff into acetyl-CoA, so with that, and then from there everything works the same no matter where you started with. Okay, so the general motif of these things is electrons for oxidation are carried by the small molecules. In this case, it's NAD and NADH. So with that, and so what can happen is this hydrogen here can go onto this reactive site. We actually talked about this in class. If you want to look at the mechanism of this, so if this is NADH, right? So drawing these hydrogens. So that's the rest of the NADH. So this is the NH2, H. So this is going to be coming in as essentially a hydride. So what's going to happen is you're going to get a new bond here. So it's going to attack here, but this carbon already has four bonds. It's got to get rid of one. Some of that. So the easiest one to do is push away these pi electrons here. So I'd really like to push those here. The problem is that carbon also has has 4 bonds, and so the easiest thing for this to do is just push it onto that carbon or that nitrogen, and so you end up with and so you end up with this. You've broken apart the aromaticity, so this thing is likely to go away. And if you want to push this away to release the hydride to be able to do something with it, you just simply reverse the reaction like that, and away, and away it goes. So put that as a hydride. Now the FAD is a little more complicated, but it runs into a very similar principle, where the here, let me draw this. So there. So this one right there. Yeah. Okay. So again, you're going to have a So again, you're going to have a hydride here, and it actually ends up going there. But again, this nitrogen wants to really only have 3 bonds, and so it's going to push these guys there, push that up there. So you're going to end up with so there was pair lone pairs there. This isn't particularly the most stable configuration for this, like that. And so what we can do is we can actually have a second hydride come in, negative charge. If that attacks there, these electrons can go there. How does this work? Oh. Yeah, that goes there. Oh, I'm missing it. I'm sorry. Miss an extra bond. That makes more sense. Okay, so this guy can come here, that guy can come there, and so you end up with this. So you end up with a hydrogen here and a hydrogen here. And so if you want to get rid of this, right, and you have the amide back and stuff like that, and so these hydrogens can go and do what you need to get done, stuff like that. And so the FAD can actually pick up two hydrides, but it still works similarly. So in general, for these things, if you use NAD or NADPH for this, NADH is typically used for the generation of ATP. NADPH is used, and the only difference is an extra phosphate down here, right there. NADPH is generally used if you want to do involve small molecules, stuff like that. So NADH is used for ATP generation. Okay, so the general motifs from these things, so activated carriers, so acetyl-CoA, stuff like that. So again, it's going to be one of those thermodynamically unstable, but kinetically stable, just like ATP. So this here is your acetyl group, okay, and stuff like that. And so this right here, this pantothenic acid, the ADP right here, this is all basically a good leaving group. Okay, so this is no different than any other sort of leaving group you learned about in organic chemistry, stuff like that. And so what you're going to end up having is simply, well, here, let me keep the colors the same. So if you had your acetyl group here, and then S, and then whatever it is, this part here, okay, all that's going to happen is you're going to have whatever your nucleophile is, negative charge, right? This is going to come there, up, down, kick away. That's it. It's just the same acyl chemistry that you learned in organic chemistry way back when, So that, and then you've got your nucleophile plus your S, S leaving group. All right, there's your CoA, stuff like that. So this is just simply CoA. It's just a leaving group. It's just a lot bigger than the ones you usually deal with. Stuff like that. So there's a bunch of different types. So activated characters, so ATP if you want to do phosphate, the flavins, if you need electrons, acyl group, aldehydes, whatever. So there's a whole bunch of these things that's used repeatedly. So it doesn't have to, so you can only need, it can provide flexibility. Okay. So remember, with these things, we're going to have oxidation reduction reactions. So if this gets oxidized, that means going this way it gets reduced, stuff like that. And so here we're reducing the number of CH bonds here. So going from 2CH2 to 2 CHs. So that's a reduction. Same thing going here. You're also, so this one's a little bit easier to see because you're going from 1 CO bond to 2, so this is an oxidate. So this is getting oxidized, something like that, and so producing this. I guess this would be oxidized as well because you're going from here. You're reducing the number of CH bonds, so this would get oxidized as well. So with that, so if these are getting oxidized, then something's getting reduced, and so this is getting reduced. So with that, so this is getting at the expense of this, which is getting oxidized. Okay. So ligation reactions, and so in this case we do redox type chemistry where we change the number of CO bonds. The other one we're going to be dealing with are ligation reactions where we're making new carbon-carbon bonds, and so in this case we're adding new CO2 molecules to this. So we're adding new carbon groups. So with that, so in this case we're adding it to this N2. So two things coming together and clicking right together. We're going to see a lot of isomerizations, and so that means a group, it looks like basically a group moves, stuff like that. And so in this case here, this OH moves from there to there, stuff like that. So you can get it folding, stuff like that. So you can get it folding differently, something like that. So it looks like it changes. So in this case, you have the OH here, like that. So glucose opening up. You've got the C double bond O, so moving from here to here. So these two groups swap. So you make the isomer of that, and then it folds back over. So we're gonna have a lot of group transfers, stuff like that. And so this is an S-adenosyl methionine. So this is basically the equivalent of methyl. This is basically another good leaving group. It's the equivalent of a bromide, stuff like that. And so you got nitrogen coming in here, doing an SN2 attack, popping this away. So with that, so just like, so it's no different than if we had NH2, right? CH3Br. So for an SN2 attack, right? So this is partial positive, partial negative. Right? It's just this wanting to make a bond there, right? The negative character of these this lone pair getting attracted to the partial character here. The carbon can only have 4 bonds, so it's got to get rid of this one. So the easiest thing to do is go there. It's no different. It's just a normal SN2 chemistry that everybody learns about in organic. So, right, plus Br minus. All right, so these electrons here become this new set of lone pairs there, and then away you go. But this is no different than S-adenosyl methionine. So, but that, so hydrolytic reactions is another type where we're adding water across it, so hydro, right? So but that, so you'll be breaking this bond, right? So when in this chorus, right, you don't want to think of water as H2O, you want to think of it as HOH. So in this case, we're adding OH on this side, H on the other, but then they can react acid-base chemistry. So you end up adding this across there. So OH goes on this side, H goes on that side. You have cleavage reactions without oxidation, stuff like that. So this is going to be particularly, this is going to be one We'll talk about this, you know, in the next chapter, where you just simply break the bond here and you add hydrogens across it. So like that. Well, well, you do get one here, so like that. So I guess you do get an oxidation there, but anyway, so like that. So you can get cleavage reaction, something like that. So, but remember, a lot of this stuff is going to get controlled. So like that, it's highly controlled. And so, and there's a couple of different ways that we can do this. Is that we can produce enzymes only when needed. So you bump up to this this high activation energy, right, where it stops. If we have too much of it, we can the cell can ramp back the amount of production that it gives. You can turn that spigot off. So like that. So the other thing is it can do is it can keep the newly formed products away from other processes. So if you don't want to make ATP and then suddenly go back and redo it, you don't want to spend all this energy making it if it can just get fed back in, because remember, the cell will recycle whatever it can, and so it needs to keep the what it just made from getting metabolized, right? So it's like buying a lottery ticket, winning $1, and then buying another lottery ticket with that, and then not winning anything. It's a net loss, right? So for that, so you're spending money and you don't want to immediately lose it. So how do we do this? So this is another way that you can do is keep it separate, so you don't burn the things you spent so long making. So that you can have allosteric feedback inhibition, allosteric control. So what you can do is you can say, okay, this product here will actually inhibit this enzyme. So if this builds up, basically it says, whoa, we don't need to make any more of this. We don't need to do this sort of reaction anymore. And so you can get this sort of non-competitive inhibition where E can come in here and block and inhibit this this enzyme here along the way. And so if it blocks it here, you don't have to spend the energy to make this you know, or that, any more of that. So by that, so it's best to get the feedback as early as you can with that, so you don't have to go through these steps unnecessarily. So so with that, so you can also do this through so you can get this allosteric effects. You can also do it with switching on and off molecules. So you can do like phosphorylation. So that can, you know, if you're low on something, it can cause the phosphorylation of an enzyme. So with that, or if you have some, you know, turn it one on, or you can if you have something build up, that'll trigger, you know, to lock the phosphate off, to flip the switch off, something like that. So you can also have it release hormones, whatever, stuff like that. And so there's a lot of different mechanisms to make it so that it doesn't make too much stuff with that. So, okay, good luck.